an earlier shift, and apparently she did quite a bit of collecting.
Oh, nice.
So
Bet
we
she
have
did.
1, 2, 2,
Yeah. Got a few sponges in here.
3, 4. I'm seeing 4 sponge samples so far.
And some of those are unknown. That's cool.
Correct. These are
Yeah,
definitely new morphologies.
Super.
They may be similar species to what we know, but the shapes are really bizarre for
Interesting.
sponges we see on the West Coast.
But they could be a new species.
They also could be their own new species.
Yeah. Now those little very branched white things that we're going by right now, those aren't corals or sponges, even though they look like they might be. Those are benthic sea cucumbers. And okay. So you would say that's a long spine, I bet, from this depth. Is that right, Erica?
Yes, and he this pectoral fin. But yes, would lean long spine.
Erica's a fish expert. I'm supposed to be a fish expert, but I'm not very good at identifying names. Mainly,
Well, real
I keep
time from
forgetting which
cameras
is in
is
which.
quite different, I imagine.
Yeah. Well, Erica, I mean, now that you guys are the pilots and the navigator and the data logger, the shift's changed and we've got all new people. So we do a intro, and Erica can tell us why she knows so much about fish in the hand from a lot of her previous experience. So should we do introductions around? What do you
see?
The
nudibranch eating that bamboo coral. It just went off screen.
Yeah.
Okay. My name is Erica Frew. I work for the Northwest Fisheries Science Center. I'm part of their AUV team. I did 10 years on the groundfish trawl survey before I switched over to a robot. So I have lots of sea time and lots of fish sampling in my backstory. So having to switch over between holding a fish and identifying it to looking at a picture of the top of a fish has been quite challenging.
Yeah, Erica, being on the trawl survey must have given you, well, I know it gave you a lot of experience looking at fish on the deck of a ship. Erica, you're breaking up a little bit. Am I breaking up when I'm talking at all?
Great, thanks.
Yeah, slightly. I can try to make some small adjustments in the V-Link system on this end. You guys were sounding perfectly clear, so it might just be a satellite thing, but let me take a look at it.
Okay. And I don't know, who do we have on navigation right now?
Yeah, so I'm Ben Gracian. I'm a navigator and mapper for Nautilus, and I'm also a PhD student at the University of Rhode Island.
Oh, that's great. Are you in Chris Roman's lab?
Yeah, I am. My brave and fearless leader.
Yes.
Yes, working with all the
we've
engineers.
worked with Chris a lot and have worked with him on his camera float.
Oh yeah, the Lagrangian float.
Yeah,
That's super neat.
Yes. And exciting, always working with Chris. And he's one of the nicest guys in the world.
Yeah, he's
so,
great.
We started working with Chris when he was a postdoc in Hanu Singh's lab in 2005 when we first started doing our AAV work.
Oh, wow.
Yep.
So, oh, that's gorgeous. Who we have on who are our pilots today? Who's on Argus?
Kim and Jess are just chatting for a sec. So
Oh, okay. I can't hear them when they chat.
Yeah.
So, just for the public, I was explaining that.
Yeah,
I think this is a really hard area to drive in because there's quite a current whipping through here.
Yeah.
Not as bad maybe as it was around midnight. I saw some of the Paragorgia were kind of flapping in the breeze, and they're pretty stiff. So for them to be shaking in the current was a little surprising.
Yeah. Hey, folks. For the introductions, my name is Jess Sandoval, part of the ROV team, and Sin Herc.
Hello and good morning, Kim Wallace, ROV team, currently sitting Argus.
Good morning.
Hi, guys.
This is Marley on the video and audio side of things.
Good morning. This is Mary Dury, and I'm the data logger.
And I guess I'm the last person to introduce myself. I'm Liz Clark. I'm at the Northwest Fisheries Science Center. I'm a senior scientist there and lead a group that is using an autonomous underwater vehicle to look at corals and sponges. But as you can hear now, I collaborate with a lot of other teams who are using ROVs to survey corals and sponges and fish. And we're really interested in where fish are and how do they use these corals and sponges. Are they habitat for them? And also we're interested in just the species diversity of corals and sponges in this area, because both are not well known, but particularly the sponges are not well known in this area. I have many other hats at the Northwest Fisheries Science Center, but my coral and sponge and fish hat is the one that's relevant right now. And we're going over some nice sponges. Most of these don't look unfamiliar to me, except some of these sponges. Well, that yellow sponge is not familiar looking to me at all. And the morphology of these sponges seems to be very kind of twisted. They look similar to sponges I've seen before, but it may be a morphology that is useful in this kind of current regime where the currents are moving really fast. Do I sound a little bit better, Erica?
But Jess has kept pretty stable. We started at 866 and you're at 872, so you've been pretty stable, Jess, on depth, which is good.
Yeah, I'm just trying to, if I go down a little, like come back up a little.
Yeah.
Labor.
This transect speed and every height looks pretty good to me, but it must be hard to do on this slope and with this current.
Yeah, so what we have the two vehicles do is we have Argus kind of go off slope a little, like in deeper water to the west. And we have Jess look a little bit more to the east so that she gets a better visual contact as we move. So her bearing and her heading are slightly off to move along the right direction, but keep a nice view for counting contact.
So those of you at home who are listening, Argus is connected directly to the ship by a tether. And you'll see that in the quad view on the top right, the view from Argus, not Argus itself. On the top right of that quad view, what you're seeing is Argus looking usually at what we call Herc or Hercules, which is on a tether connected to Argus. That's sort of that way of using two ROVs kind of decouples the ROV that's nearest to the bottom, Herc, from the movement of the ship. There's other advantages too. You can kind of see what the Hercules is doing from Argus, and what are the other advantages of a two-body system, as they call it here? Is that a Petrali? I'm sorry, Erica, is that a Petrali that just went out of view?
I missed it, but I'm guessing it this depth probably not.
Sorry. What's the other advantage of having a two-body system?
Raj, yeah, we use a lot of the two-body system is also quite good for. Sorry, we're doing a little bit of ops thing. So if I go over the same space as what you've just mentioned, apologies. But yeah, so with a two-body system, well, Argus kind of acts as a weight to disassociate from the ship, but there's also other kind of free flyers that they'll also do this type of either catenary with the tether in the water, things like this. So, but Argus is really useful for one, the visual contact and visual surveys, so we have another kind of perspective when we're flying through the water. And two, also for all the sensors that are on board, so we can kind of get a better picture of sonar wall or that our sonar targets and hits of walls and things like this. So it's very useful for that as well, as kind of getting more of a spatial realization of where you're at in the water, a very good disassociation from the ship, and yeah, and good sonar hits and targets. So that's at least one big advantage that we have with having Argus in the water as opposed to just a weight.
Yeah, thank you.
Sure.
What do you think about those morphologies of those sponges, Erica, that look like catcher's mitts? That's what I'm going to call them.
The platy ones, I'm seeing some mycale in there. And it looks like Abby grabbed a sample of one earlier, so maybe in a little while we should grab a second one just so she has something to compare.
Sure. Ben, what's the distance we've
Yeah.
on this
How many sampling boxes do we have open?
Like 4 or 5 meters left.
4 or 5, Raj. Let me see. Mary, I don't think you're on SPL. Microphone's too far.
We have 3 starboard bio boxes and 3 slurps.
Okay. And for those of you at home, those are the boxes that are on the Hercules that we put samples in, and they also have a slurper, which I hope you'll be able to see us use to take some samples.
Yeah.
They take other samples. There's manipulators on the ROV as well. And are the coral cutters on?
Yes, they are.
And a cutter on the manipulator for picking up samples as well. Erica, what's the depth range typically of petrale? The reason I was hoping that was petrale is this sort of broken up sedimented rock is typical of the area. We saw a lot of petrale in much shallower water.
Yeah, they're usually much shallower, in the 200 to 300 meter range, or 200 to 400 meter range.
Typically in this depth, you'll see Dover sole and deep sea sole, I would say, but it didn't look like either of those. What other flatfish would you see down here, Erica?
Down here would be deep sea sole, Dover sole, the occasional Rex. They're the ones that go the deepest.
So typically on these dives for the public, we would be on the ship, but because of the current pandemic situation, we've got a very limited group of scientists, not limited in their skills, but limited in their numbers on the ship. And so we are talking to you from Erica's office, home office, and me, you're talking to me from my dining room. And then we have people on chat who are in a chat room, who are other scientists who are watching what we're doing and helping us out with identifications or suggestions on sampling. And our coral expert has just, I think, oh no, Meredith is still on, I think. He's sticking with us, but Lisa Wunick, who is from the Sanctuary Program on the West Coast, that's a group of national marine sanctuaries on the West Coast, and she's in the regional office there, and the co-lead with her is Lizzie Duncan from the Channel Islands National Marine Sanctuary. But Lisa's just signed off. She was on the last watch, and she's going to take a nap for a while before she has to be on watch again. And you'll be hearing Lizzie, I think, later in the day from the Channel Islands National Marine Sanctuary. We're from the Northwest Fisheries Science Center of NOAA, and we have collaborators from the Southwest Fisheries Science Center as well. Guy Waters, I think, was on the 8 to 12 watch today, and I think Tom will be.
We have come 200 meters on this transect, so I think we can call that.
Roger that.
Great. Thank you.
And
So, Erica?
yeah. So I see this kind of arbitrary target, if you can look in HiPAC, that was made mostly to the north of us, slightly downslope, but still on this cliff. So we can continue heading there and then come off slope after, at that point. But does that sound good? We continue along this steeper portion?
Sounds good to me.
Yeah, sounds good to me. I'd like to get up on that edge eventually and just a little bit over after we hit that waypoint.
Roger. So I'll just keep on the contour and then we can kind of have access to that slope for move up it.
Sure.
Okay.
So when Liz, when you say at the edge, you mean over at that waypoint drop there?
Yeah.
Okay, great.
At the 10:30:08? Or
Yeah.
is it a different? Okay, perfect.
Okay.
Does it sound okay to you, Erica?
Yeah, I'm just trying to relay what Lisa and Meredith had set up for us, and Guy Cochran's been on for the last shift, so.
All right, I'm gonna bring us to, let's see what you're, yeah, let's keep on 345 for a bit.
Roger.
Oh, right close to the screen there. Looks like a little snailfish. Am I right, Erica?
I've got the map up. Hold on.
It went by. It's not on there anymore.
Do these benthic not versions, the wrong word, species of snailfish, do they all have the modified pelvic and pectoral fins to form these like pelvic sucking discs or suction discs?
They do not have the disc,
Okay.
like a.
So only the intertidal ones are the ones with the suction discs.
Right. Yeah, they don't have the same adaptation.
Roger. Interesting.
So Jess has no interest in them anymore.
They're very cool though. They're usually pink and they have blue eyes. They have other features.
Redeeming colors.
Yes. Or different shapes. Some of them look like almost like lollipops. Their heads are very large and round, and their tail and body are very skinny.
Interesting.
They are one of my favorite fish because they're up close. They're kind of adorable with their little chubby cheeks. And down here, there's a lot of really kind of hot pink ones. And I'm going to say that's a Dover sole there. Am I right, Erica?
Correct. Yep. Is that the same one you saw earlier?
No. The other one looked to me, and again, it was just on the edge of the screen. It looked wider and rounder, which made me think it was not a Dover. But I only saw it on the edge. And it was wishful thinking on my part. Can we zoom in on that coral right there in the center of the screen?
The mushroom
to
coral
give-
there?
Yeah.
Sure.
Just for people at home so they can
All right,
see
Marley, go ahead.
how great looking those are up close.
Yeah, these are always cool.
And there's a plate sponge there also, just below it. And that's a,
Sorry,
I can
come a
never
little
say
wider,
the,
Marley.
a hetero, I can never say the name, Erica. You want to pronounce the name of that sponge for me?
Heteropolypus ritteri.
That's it.
All right, you want to do a quick zoom there again? Try to get us stable.
Current knocking you around.
WDFW has the range of Petrale slowed down to 550 meters.
Go soon, but
Bridge nav.
There we go. Nice.
So do the juveniles and adults tend to have the same kind of zonation, or is there kind of a differentiation over the life history once they settle?
all right, Marley, go and come wide, please.
They, excuse me, the juveniles do stay more shallow, but I think what Liz is looking for is the adults tend to migrate to shallower water for spawning events
Oh, I see.
and spawning aggregations.
There's a deep sea slough, the really fancy looking slough up there on the right hand side we just went by.
So we've seen large petrale aggregate. You'll have one female with several males surrounding her, and this happens usually later in the year, and it's actually what a lot of the commercial fisheries kind of target when they can catch a lot of petrale in one place and time.
We'll be up, I think, doing a dive potentially on the site where we typically have seen those spawning petrale. I think the last dive before we go down to the Channel Islands National Marine Sanctuary.
Oh, cool.
I haven't looked at my email this evening, but we may be planning a dive to do that, to look at that area. There's a dead sponge going by on the right. You can tell it's a sponge in trouble because it's covered in sediment.
A sponge in trouble. Sounds like a novel.
So it looks like this the steep feature is going to level out as we go north, but kind of towards the target that we made, it's steeper.
Mm-hmm.
So I don't know if science, if you want to start heading down slope now so that we can stay on the steepest feature as we go north. See, there's a little. Or we can stay on this contour and just jet west once we're there. Have the slope map in HiPAC up.
Erica, what do you think?
From what I understood, they want to stay steep.
Roger.
Okay.
And you said you wanted to go up on top of the ridge too, right?
Yeah, I think what I understood Lisa to say is, and Meredith, and maybe they can chime in here if Meredith's still on, it's right at the edge of where it gets really slopey is where they saw very high densities of coral there. And let's see if Meredith is still on.
Yeah. Let's see.
Meredith, can you chime in on the science chat and tell me exactly the zone where you saw the highest densities and diversity of corals?
Yeah, so if you stay on this contour, you'll...
Go ahead.
Yeah, so just if you stay on this contour, you will keep closer to the, to kind of summiting a little, but it does look like it gets steeper towards the base on the west. So, you know, both sound like good options. I just wanted...
Okay, Guy Cochran just chimed in and he would like us to go up a couple of contours.
Yeah.
Up.
Up the steep.
Okay, so that
I
will be
believe is what he's-
downslope of us. That makes sense. Do you see?
Go up a couple contours? Do you mean up, going up and
No, upslope.
upslope, right?
Okay, Roger.
So we'll be
So,
going
yeah,
upslope
so let's
as
just
well.
keep going north, and that
Okay.
will bring us over.
Would you mind showing the, not the slope map?
The backscatter?
Back,
Just regular.
no, not backscatter, sorry, just, let me go this.
Okay, Guy's saying we're below the steep area again and he'd like to see some northeast.
Okay, yeah. So locally I can go northeast. If you look at the HYPACK map, for the next 400 meters or so latitudinally, the steepest feature is downslope of us. But I'll bring us over, just keep north and northeast to go up. That works for you here.
Okay, that's just the direction we're getting, so
Roger.
that sounds reasonable.
Cool.
And then I guess we'll end up east of the point and then come back west over it, possibly, or?
Yeah, that sounds good. That point is...
Okay, okay.
Sounds good. But what are you thinking for your move?
North won't get a, okay, so
I'm going to do 010.
010, Raj.
Or I want to stay on it for as long as possible, but that will be good. All right.
All right, 010.
So
Yep.
Guy's saying that
010.
north won't get us out of this.
Roger. Okay, so we can just go upslope completely. It will only be about 70 meters or 80 meters, but let's do that.
Yeah, why don't we do that?
Okay.
So that'd be more of like a
Easterly.
0600
Yeah, exactly. I'm thinking
80.
let's do 065.
065, Raj. Zero 70, Roger.
For those of you at home, you can see those two little dots there are the lasers, which we use to measure things or get a relative size of things. And those lasers are set, I think, 10 centimeters apart. And they are on the Hercules. You can see those two little green dots really well right now.
That'll be an elevation change of 40 meters or 30 meters.
Yeah, it's going to be 20, 30 meters immediately. I mean, those are the steep parts, will be 20 or 30.
Roger that.
Yep.
So once we're to the east, once we're directly upslope, do you guys want us to follow that contour along until we're directly west of the waypoint or of the 10, 30, 50 waypoint, and then drop down kind of downslope, or would you rather we just kind of make a beeline there? A beeline being a direct line. Sorry, that wasn't clear.
Yeah, yeah, I wasn't sure how firm that target was.
Roger.
So if you guys want to adjust again for that steep feature that's downslope of us after this.
Okay, waiting to see if Guy chimes in on this one.
Yeah, roger.
Roger that.
Okay, can you zoom back out so we can see
I think she wants
on
to zoom out on high pack.
high pack?
Oh, yeah. Sorry.
The V-Link seems to be a little bit off and on right now.
Mostly on.
Mostly on.
Mostly on. Okay, so 3F is just coming back into view off the edge there. Okay, guys saying head for 3F when you get to where you'd go over that steep area on the way to 3F.
Sorry, can you repeat that?
I'm trying to make sense of it
Yeah.
myself.
Yeah, because again, you know, F is going to be way across this flat feature, so that steep part on our side is really only taken advantage of if we can attack it from east to west, pretty much. So, yeah, but we can definitely try to get over it as we get to F and see how much visual contact.
Roger that.
Yeah.
So continue northeast a bit and then come all the way east to west down that slope area on the way to F, I'm guessing.
Sure. Sounds good. So it seems like we're going to be going down slope, and then just drop and move.
Okay.
Might get a little bit of a.
Yeah, right now we're just going directly upslope, so we'll be off the slope probably in about 90 meters. Then we can go directly west. That's my understanding.
Yeah. West to deeper waters.
Yep.
Roger that.
Thanks, guys,
Yep.
for being flexible.
Of course.
You can't tell if these red plexaurids are happy or not happy. Some of them are looking pretty spindly.
What's the process that would be going on? Are they actually, would they be losing soft tissue?
I don't know. They all seem to have, I don't know if they're symbionts or hitchhikers or what hanging off of them. You we saw a nudibranch earlier eating a bamboo coral, so there could be some predation involved. I'm not sure.
Yeah. But spindly's not good.
Yeah, almost. They're almost sloughing off some of their soft tissue, so it looks like some open spots on their skeletons.
Got it.
Like that one right above the lasers now. Looks.
Marley, want to push in, please?
Pretty brittle. Oh yeah, ophiuroids all over it.
So that's a paragorgia in the bottom right.
Ah, sorry guys.
That one, yeah. So maybe it's just polyps that make them look fuzzy rather than.
Come wide, Marley. Sorry guys.
Damage.
But corals in better conditions or good conditions will grow a thicker, stronger, more calcified skeleton, or
I'm not sure. These are quite long and thin for a plexaurid, and they seem to be falling over more, I guess. So I don't know if they're overgrowing their structural integrity or what.
right?
It's a cool sponge. Marley,
Oh,
just
it's a
snip
very
there.
cool sponge.
And a little baby mushroom coral.
All right, come wide, please, Marley.
It's a chitin on the rock behind that, and thorny heads. And that one actually had his dorsal spine up, which makes it much easier to identify. That's how you tell the difference between a short spine and a long spine is the length of their third dorsal spine.
Roger, thanks.
Several bamboo corals coming up. It looks like a small heterocone base sponge in the center of the screen. Seeing Paragorgia, lots of the mushroom corals.
Oh, it's a big perch. Oh, an escape.
What's a larger short spine thorny head? They get much bigger than the long spines.
Really see inside it's a perculum.
Yeah. The fishermen, commercial fishermen would call them bucket heads.
Oh, yeah. Definitely looks that
And
way.
that's a rough-tail skate. Yeah, they have these little tiny bodies with these giant heads. And they're ambush predators, so they have a huge gape. There's another snailfish coming through.
Yeah, it seems advantageous to have that. Seems like this
I've
scorp.
seen I've seen short spines like that come up with the big grooved tanner crabs in their stomachs.
Wow. Because even that snailfish is a scorpainid, right? They seem to be super successful down here. That whole family or order. Scorpainid.
Ooh.
All right.
Looks like you're leveling off a bit here.
Yeah. Which actually it's pretty precise with the map, which is nice. Yeah, there's a little, but let's just finish the ship move. It's another 15 meters and get you to the end of the tether so we can see the end of this. And then again, I don't know if Guy, we can just go west, but if he wants to go a little to target just the bottom of that steeper slope, I just don't want to make it too difficult for the positioning of Argus. So.
No worries. Yeah,
Okay.
that should be fine.
Great. Yeah, I'll try to maximize that.
What we'll do is we'll just turn Argus to the heading and then make sure we're off the deck 20 meters, and then
Yeah.
I'll just kind of turn and face the slope and go down.
Roger. Sounds good.
Okay, guys chiming in. This is the top. We should go to the southeast part of the steep area.
Bridge nav, you.
And he's agreeing with your assessment, Ben. So.
Okay, cool. Not to be too chatty, I just want to get good data for you guys.
Nope. We want to see this. We want to see the steep, but
yes.
we want you guys to be comfortable with it.
No,
so.
we're good. All right, so let's see. So, Jess, I'm thinking, I'm thinking like 285 will be good for us.
285, Roger.
Okay.
285.
Just saw a McTophid or a Bathylagid drive through. The pom-pom anemones are fun. Liponema. Guys asking if we could put a target on the nav for where we're going.
Yeah, I have it up. It's called Temp Bottom Slope.
Awesome.
Yep. Actually called Temp
You
Bottom
are,
Slope. I can change that.
you are all over this. There's
Oh
another,
yeah.
or is that the same skate we came over laying next to the thorny head?
Looks like it, yeah. This current is just pushing us that way.
I can also slow down to 0.25 or 0.2 if you need.
Yeah, we'll see how we do, but it looks like I'm getting pushed around by the current.
Yep.
Oh, sorry, Kim. Roger.
Seeing lots of Garsemia corals. Again, Paragorgia and bamboos. Mushroom corals, Heteropolypus. Long spine thorny heads. It's a bigger short spine there. Another way to tell them apart, if you have the fish in your hand, is to give them a little squeeze and have them open their mouths. And the inside of a short spine is pure white, and a long spine will have black on the inside of their mouths.
Must be much easier.
When we were up in Olympic Coast National Marine Sanctuary, we were trying to get a picture of a yellowmouth rockfish because there are no guides that have a picture of the yellowmouth in situ.
I read that in the dive plan. I was like, that's really opportunistic.
So we tried to have her ask the rockfish to open their mouth. Needless to say, they did not comply.
Yeah, this is fine. If anything, it'll just, if I get too behind, then I'll just blow off the slope, so it's good.
Roger.
We need to come up with some sort of bait system. Maybe that would help. Another short spine of the lasers.
Very pretty terrain.
Yes. Bamboo corals, and are we in a place where we could zoom?
Yeah, very briefly.
Okay,
Marlene,
over
to do a
the
partial.
right.
Sure.
Oh, sorry, was this not it?
Oh.
Right. No, that's all right. That's all right.
Okay.
Zoom in, yeah, or
Yeah, a little more. All right, a little more. Sorry, Marley.
that's all good.
All right, go ahead and come wide, Marley, please.
Stripped of polyps, so you can see why they're called bamboo coral. They have segments. Looks like a scarlet king crab up there. Either a lithodes or a neolithodes.
Do you know if those red spots on the bottom are polychaete tubes or something? I just saw one of them retract.
I believe they are some sort of tube worm, yeah. Because they're quick.
Yeah.
We also come across some ground-dwelling anemones
Oh.
that'll do the same thing, retract quickly, but they're usually more in sandier areas. Softer substrate for sure.
Yeah, I guess it would be hard to bury here.